Welcome to our channel. We are so glad you found your way here. Let us help you fall asleep. Let's begin with DNA packaging, the impossible geometry of your genetic code. Take a slow, deep breath in and release it. Let the weight of the day fade away as we embark on an unhurried, atmospheric journey through the wonders of the cosmos. Your mind is quiet, the universe is vast, and we are ready to explore. This exploration into DNA packaging reveals an intricate, three-dimensional architecture that sustains life within every cell. The human genetic code, comprised of 3 billion base pairs, must fit into a microscopic nucleus. This remarkable feat is accomplished through a highly organized system of folding and condensing, transforming lengthy strands into compact structures. The double helix unwinds, revealing its sequences, which interact with specialized proteins known as histones. These proteins play a pivotal role, acting as spools around which the DNA winds, forming nucleosomes. Each nucleosome resembles a bead on a string, allowing for further compaction through higher-order structures. This region of genetic material, wrapped and folded, creates a hierarchy of organization. Various levels of packaging contribute to the accessibility of genes, determining whether they are active or silent. Influenced by cellular signals and environmental factors, the organization is not static. It undergoes dynamic changes throughout the cell cycle, particularly during replication and transcription phases. Intriguingly, this architecture is not uniform. Different cell types exhibit unique patterns of DNA organization, reflecting their specific functions and requirements. Such diversity underscores the importance of spatial arrangement in regulating gene expression. Understanding this structural complexity can provide insights into genetic disorders and innovations in gene therapy, offering potential pathways for future research. This structural complexity reveals itself most prominently in the iconic double helix formation. This arrangement consists of two long strands of nucleotides twisted around one another, resembling a twisted ladder. Each strand is composed of a sugar-phosphate backbone with nitrogenous bases paired in the center. Adenine pairs with thymine, while cytosine pairs with guanine, creating a specific and highly regulated bonding system. The helical structure is stabilized by hydrogen bonds between these bases, contributing to the overall stability and integrity of the molecule. This configuration is not merely aesthetic, it is a functional necessity. The double helix facilitates the precise replication of DNA, ensuring that genetic information is accurately passed from one generation to the next. In these spirals, the sequence of bases encodes the instructions for building proteins, essential to cellular function and organismal development. The helical nature also allows for compact packaging within the confines of the nucleus, organizing the vast lengths of genetic material efficiently. Variations in the geometry of the double helix can occur, influenced by environmental factors and specific nucleotide sequences. These variations may lead to different forms of DNA, such as aDNA and zDNA, which possess unique structural properties and biological implications. Each form contributes to the overarching complexity of genetic expression and regulation. Nucleotides serve as the fundamental building blocks of deoxyribonucleic acid, comprising a sugar, phosphate group, and nitrogenous base. This intricate structure allows them to link together, forming long chains that, in this region, the arrangement of nucleotides influences various biological processes. The precise order determines not only the proteins synthesized but also their function and regulation. Variations in these sequences can lead to significant differences in traits and characteristics among organisms. This molecular arrangement is tightly regulated, allowing cells to respond to environmental cues and maintain homeostasis. The role of nucleotides extends beyond mere structural components. They participate in critical cellular processes such as energy transfer and signal transduction. Adenosine triphosphate, a nucleotide, serves as the primary energy currency of the cell, facilitating biochemical reactions necessary for life. This dynamic interaction of nucleotides highlights their dual role in both genetic information storage and cellular functionality, emphasizing the complexity underlying the simplest aspects of biological existence. Chromatin serves as the intricate DNA-protein complex that organizes and regulates genetic material within the nucleus. Composed primarily of DNA wrapped around histone proteins, this structure plays a vital role in managing the accessibility of genetic information. The packaging of DNA into chromatin allows for efficient storage, enabling an astonishing amount of genetic material to fit within the microscopic confines of a cell's nucleus. This complex exhibits various forms, existing in both condensed and relaxed states. In its compact form, chromatin assumes a higher order, facilitating the segregation of chromosomes during cell division. Conversely, the more relaxed configuration permits essential processes such as transcription and replication, where genes are expressed or duplicated. The balance between these states is critical, influencing cellular identity and function. Post-translational modifications to histones further diversify chromatin structure, creating a dynamic environment responsive to the cellular context. These modifications can signal whether specific regions of DNA are active or silent, underpinning cellular differentiation and adaptation. This layered complexity of chromatin not only underscores the remarkable efficiency of genetic organization, but also reveals the intricate regulatory networks that govern gene expression. Understanding these mechanisms offers profound insights into the fundamental processes that sustain life and the potential implications for disease treatment and biotechnology. Histones play a critical role in the organization of DNA within the nucleus. These proteins serve as the structural framework around which DNA wraps, creating a complex known as chromatin. This interaction transforms the long, linear strands of genetic material into a highly compacted, manageable form. Each histone core, composed of specialized amino acids, forms an octamer, around which approximately 147 base pairs of DNA coil. The resulting chromatin is not merely a passive storage system. It actively participates in regulating gene expression, determining which genes are accessible for transcription and which remain repressed. This regulation is influenced by various chemical modifications to histones, such as acetylation and methylation. These modifications can alter the affinity between histones and DNA, subsequently affecting the chromatin's structure and function. This intricate system allows for a dynamic response to cellular signals, dictating cellular behavior and identity. Specific patterns of histone modification are recognized by other proteins, which mediate the recruitment of transcriptional machinery. The implications of this regulation extend beyond mere genetic control. Understanding histone dynamics is crucial in elucidating the mechanisms underlying various diseases, including cancer. Research continues to explore how manipulation of these proteins might offer novel therapeutic avenues. The complexity of this interplay reflects the fundamental elegance of biological systems. Nucleosomes serve as the fundamental units of DNA packaging, organizing genetic material in a manner that balances accessibility and protection. Each nucleosome comprises a segment of DNA wrapped around a core of histone proteins, forming a structure reminiscent of beads on a string. This arrangement allows for efficient compaction of the genomic content, ensuring that vast amounts of information fit within the confines of the cell nucleus. The wrap of DNA around histones is not merely structural, it is integral to the regulation of gene expression. Modifications to the histone proteins can alter the interaction between DNA and these proteins, influencing the accessibility of specific genes. This dynamic modification landscape plays a pivotal role in cellular differentiation and response to environmental cues. In eukaryotic organisms, the formation of nucleosomes leads to higher-order structures, such as the 30-nanometer fiber, further compacting the genetic material. This hierarchical organization is crucial for maintaining the integrity of the genome during processes such as replication and transcription. Nucleosomes also facilitate the orchestration of complex regulatory networks, impacting cellular functions across diverse biological contexts. As they undergo various modifications, they contribute to the epigenetic landscape, influencing not only individual cells, but also the organism as a whole. The intricate design of these fundamental units exemplifies the remarkable adaptability of life itself. This remarkable adaptability is further illustrated by the formation of the 30-nanometer fiber, a critical higher-order structure of chromatin. At this scale, nucleosomes, the basic units of DNA packaging, coil together to create a dense, organized fiber. This configuration allows for the efficient storage of genetic material, accommodating the extensive length of DNA found within each cell. The 30-nanometer fiber emerges through two primary models, the solenoid and the zigzag. In the solenoid model, nucleosomes wrap around one another in a spiral formation, resulting in a compact structure. The zigzag model, in contrast, presents a more linear arrangement, allowing for varying degrees of accessibility to the underlying genetic information. Both structures highlight the dynamic nature of chromatin and its capacity to regulate gene expression effectively. This level of organization is essential for cellular function, as it facilitates the proper segregation of chromosomes during cell division. The interaction between this structure and various regulatory proteins allows the cell to respond to environmental changes and developmental signals. Each conformation, whether tightly packed or more relaxed, plays a pivotal role in determining which genes are activated or silenced. Supercoiling serves as a crucial mechanism in the organization of DNA, influencing both its physical properties and biological functions. This phenomenon arises from the twisting of the double helix, resulting in a state where the strands become intertwined and compacted. Such configurations facilitate the efficient packaging of genetic material within the confines of cellular structures. Negative supercoiling, characterized by the unwinding of the helix, promotes the accessibility of specific regions of DNA, enabling the binding of transcription factors and other regulatory proteins. This accessibility is vital for gene expression, allowing for the rapid response to environmental signals. In contrast, positive supercoiling can hinder access to these regions, thereby silencing certain genes. The interplay between supercoiling and various enzymes, such as topoisomerases, ensures that the DNA remains in an optimal configuration for cellular processes. These enzymes can introduce or remove supercoils, thus dynamically regulating the organization of the genetic material. This region of DNA, with its intricate architectural features, reveals the delicate balance between stability and flexibility. The geometric complexities inherent in supercoiling underscore its significance in maintaining the integrity of genetic information, while facilitating the necessary adaptability for cellular function. The ability to navigate these structural challenges is fundamental to the survival of all living organisms. Euchromatin and heterochromatin represent distinct forms of chromatin, each playing a vital role in the organization and functionality of genetic material. Euchromatin, characterized by its loosely packed structure, facilitates active transcription processes. In this open configuration, genes are readily accessible, allowing for the expression necessary for cellular function and adaptability. The dynamic nature of euchromatin enables it to respond swiftly to the needs of the cell, engaging in various cellular activities as required. Contrastingly, heterochromatin exhibits a tightly packed conformation, which serves to maintain structural integrity and protect genetic information. This compact form is often associated with gene silencing, providing a regulatory mechanism that prevents the expression of certain genes at specific times. Within heterochromatin, the regions of DNA are less accessible to transcription machinery, ensuring that essential processes do not inadvertently activate. Both euchromatin and heterochromatin exist within the nucleus of eukaryotic cells, contributing to the overall architecture of chromatin. This division is not merely a structural distinction, but reflects the underlying regulatory complexities that govern gene expression. The balance between these two types of chromatin is crucial, as it directly influences cellular identity and functionality. The interactions and transitions between euchromatin and heterochromatin illustrate the intricate dance of genetic regulation, essential for the organism's development and response to environmental stimuli. This complexity becomes particularly evident during cell division. As a cell prepares to divide, the packaging of DNA undergoes significant reorganization. Chromatin condenses into tightly coiled structures known as chromosomes. This transformation is critical for ensuring accurate segregation of genetic material. Each chromosome, now discernible under a microscope, contains the genetic information necessary for the development of a new organisms. In this tightly regulated process, enzymes and structural proteins play vital roles. They orchestrate the compaction of DNA, ensuring that it is not only accessible for replication and transcription, but also adequately protected against damage. This careful arrangement minimizes the risk of errors that could lead to mutations or chromosomal abnormalities. The significance of this process extends beyond mere organization. Proper DNA packaging is essential for maintaining genomic stability throughout the cell cycle. Mistakes during this phase can result in aneuploidy, a condition where cells possess an abnormal number of chromosomes, often leading to serious developmental issues or diseases such as cancer. Furthermore, the interplay of various chromatin modifications influences gene expression patterns, impacting the fate of daughter cells. Thus, the intricate mechanisms governing DNA packaging during cell division are foundational to life, shaping not only individual cells, but entire organisms. Each step within this process reflects the underlying complexity of genetic information management. Interphase chromatin plays a crucial role in gene regulation, providing a dynamic framework for the accessibility of genetic material. Within this phase of the cell cycle, chromatin undergoes significant modifications, allowing for the expression or silencing of specific genes. This region is characterized by a delicate balance between condensed and relaxed states, enabling the cellular machinery to interact with DNA when necessary. The organization of chromatin influences gene activity, as regions of tightly packed DNA are generally inactive, while more loosely organized sections promote active transcription. Proteins known as transcription factors bind to these accessible areas, facilitating the initiation of gene expression. This interaction is finely tuned, responding to various signals from the cellular environment, ensuring that genes are expressed at the right time and in appropriate amounts. Changes in the structural configuration of interphase chromatin are driven by several mechanisms, including chemical modifications to histones and the presence of non-coding RNAs. These modifications act as signals, determining the recruitment of additional proteins that further influence chromatin state. As a result, this intricate system serves as a regulatory hub, coordinating cellular responses and adaptations. Understanding the role of interphase chromatin in gene regulation reveals the complexity of cellular identity, where each cell's function is dictated by the specific genes it chooses to express. The implications of such regulation extend far beyond the individual cell, impacting entire organisms. The structural differences in DNA packaging present a fascinating contrast between prokaryotes and eukaryotes. In prokaryotic cells, which lack a defined nucleus, DNA resides in a region called the nucleoid. This region often contains a single, circular chromosome, compacted and organized through supercoiling. Proteins such as histones are largely absent, though specific nucleoid-associated proteins play critical roles in maintaining the integrity and organization of this genetic material. Eukaryotic cells, on the other hand, exhibit a more complex arrangement. Their DNA is linear and contained within a membrane-bound nucleus. The packaging involves multiple layers of organization, beginning with the wrapping of DNA around histone proteins to form nucleosomes. This structure further coils and folds into higher-order formations. These differences in DNA organization are not merely structural. They have profound implications for cellular processes. Eukaryotic cells can regulate gene expression with greater precision, allowing for diverse cell types and functions within multicellular organisms. Prokaryotic cells, while simpler, exhibit remarkable adaptability and rapid response to environmental changes. The evolution of these distinct strategies highlights the intricate relationship between structure and function in the realm of genetics. The nuclear envelope serves as a critical barrier, regulating the accessibility of DNA within the cellular environment. This double membrane structure encapsulates the nucleus, providing a selective gateway for molecular interactions. Its composition, primarily composed of lipid bilayers and associated proteins, facilitates the transport of essential molecules while maintaining the integrity of the genetic material housed within. Inside this enclosure, chromatin exists in a dynamic state, compacting and expanding in response to various signals. The structural organization of chromatin is pivotal, as it dictates the accessibility of specific genes, influencing their expression. Nuclear pores embedded in the envelope act as conduits, allowing proteins and RNA to traverse the barrier while simultaneously safeguarding the delicate DNA strands from potential disruptions. The spatial arrangement of chromatin within the nucleus further underscores the complexity of genetic regulation. Regions of active transcription exhibit a distinct positioning, often found near nuclear pores, which enhances their accessibility. Conversely, regions of silent genes are sequestered in less accessible areas, emphasizing the importance of spatial dynamics in gene expression. This compartmentalization and regulation underscore the sophisticated mechanisms that govern genetic activity. The nuclear envelope, while acting as a protective barrier, simultaneously orchestrates a finely tuned environment for the precise regulation of genetic information. As a result, this structure plays an essential role in the overall functionality of cellular processes. Non-coding RNA significantly influences chromatin structure, contributing to the regulation of gene expression and maintaining cellular identity. These molecules, despite not coding for proteins, exert profound effects by interacting with chromatin and associated proteins. They are involved in the formation of various RNA-protein complexes that modulate the accessibility of DNA. In this region, non-coding RNA can facilitate the recruitment of chromatin remodeling complexes, which alter the positioning of nucleosomes. This alteration is crucial for regulating transcriptional activity, as it determines which genes are accessible for expression. The presence of these RNA molecules can lead to the establishment of specific chromatin states, including heterochromatin and euchromatin, each with distinct functional implications for cellular behavior. Certain non-coding RNAs also play a role in the formation of transcriptional domains, where groups of genes are co-regulated. Through these mechanisms, they help orchestrate cellular responses to environmental stimuli and developmental cues. The intricate balance of these interactions is essential for the stability and dynamic nature of chromatin, further emphasizing the complexity of genetic regulation. This multifaceted involvement of non-coding RNA highlights the sophisticated layers of gene regulation, showcasing how seemingly simple molecules contribute to the intricate architecture of life itself. The mechanics of accessibility within chromatin reside in specialized formations known as chromatin remodeling complexes. These complexes act as molecular machines, altering the structure of chromatin to expose or obscure specific regions of DNA. By repositioning nucleosomes, they facilitate the binding of regulatory proteins and transcription factors, orchestrating critical processes such as gene expression and DNA repair. This transformative capability hinges on the energy derived from adenosine triphosphate, a molecule that fuels these complexes. The consumption of energy enables them to slide, evict, or restructure nucleosomes. Such actions are pivotal in responding to cellular signals and environmental cues, thus allowing the genome to adapt to various biological demands. Different remodeling complexes exhibit distinct mechanisms and regulatory roles. Some promote accessibility by loosening the tightly packed chromatin, while others may compact it to repress transcription. These nuanced actions create a dynamic landscape where information encoded in the genetic sequence can be selectively accessed or silenced. In addition to their immediate effects, chromatin remodeling complexes play a role in long-term genomic organization and stability. They influence higher-order chromatin structure, which is crucial for maintaining the integrity of the genome during cell division. This intricate network of interactions underscores the essential role of remodeling complexes in the overarching narrative of genetic regulation. Epigenetic modifications serve as a critical layer of information that influences how genetic material is expressed. These chemical alterations do not change the underlying DNA sequence. Rather, they impact the accessibility of specific genes. Methylation, for instance, involves the addition of a methyl group to DNA, typically resulting in gene silencing. In contrast, acetylation of histonase facilitates a more open chromatin structure, promoting transcriptional activity. This duality illustrates a finely tuned regulatory mechanism. Regions of DNA that are heavily methylated are often found in areas associated with developmental genes, effectively turning them off during specific cellular functions. Conversely, active genes are frequently found in regions where histonase are acetylated. The interplay between these modifications creates a dynamic landscape, allowing cells to respond to environmental stimuli and developmental cues. The implications of these modifications extend beyond individual cells. They contribute to the heritable traits observed in organisms, as epigenetic marks can be passed down generations. This phenomenon reveals a complex relationship between genetics and the environment, suggesting that experiences can permanently influence gene expression. Understanding this chemical code provides insight into numerous biological processes and diseases, including cancer, where aberrant epigenetic changes can lead to uncontrolled cell growth. Thus, the study of epigenetics stands at the frontier of molecular biology, revealing the profound complexities of genetic regulation. Methylation plays a crucial role in gene expression, acting as a key mechanism in the regulation of genetic activity. This process involves the addition of a methyl group, a simple chemical unit comprising one carbon atom and 3 hydrogen atoms, to specific sites on the DNA molecule. Such modifications can drastically alter how genes are expressed without changing the underlying DNA sequence. In this context, methylation often occurs at cytosine bases in regions known as CpG islands. When methyl groups are attached to these sites, they can inhibit the transcription machinery from accessing the DNA, thereby silencing the associated gene. This selective silencing is not random. It often correlates with developmental stages and environmental influences, underscoring the dynamic nature of genetic expression. The patterns of methylation are influenced by various factors, including age, diet, and exposure to environmental toxins. As a result, this regulatory mechanism serves as a bridge between the genotype and phenotype, illustrating how external factors can shape genetic expression. Research continues to uncover the intricacies of methylation patterns and their implications for health and disease. Dysregulation of methylation has been linked to numerous conditions, ranging from cancer to neurological disorders, highlighting its significance in understanding biological processes and potential therapeutic avenues. The ongoing investigation into these modifications reveals the elegance and complexity inherent in genetic regulation. Acetylation plays a pivotal role in modulating chromatin dynamics, influencing the accessibility of DNA for transcription and replication. This chemical modification occurs primarily on lysine residues of histone proteins. The addition of acetyl groups neutralizes the positive charge of histones, resulting in a more relaxed chromatin structure. Such alterations create an environment conducive to the binding of transcription factors and the transcriptional machinery, which facilitates a transition from tightly packed heterochromatin to a more open euchromatin configuration. This shift is crucial for gene expression, allowing necessary genes to be accessed when required. Conversely, deacetylation leads to a more compact chromatin state, effectively silencing gene expression. Various enzymes govern these modifications. Histone acetyltransferases are responsible for adding acetyl groups, while histone deacetylases remove them. Their balanced activity is essential for maintaining cellular homeostasis. Dysregulation of these enzymes can lead to aberrant gene expression patterns, contributing to various diseases, including cancer. In this intricate landscape, acetylation not only serves as a marker for active transcription, but also influences higher-order chromatin structures. Understanding these dynamics offers insights into how genetic information is organized and expressed within the confines of the cell nucleus, reflecting the delicate interplay between structure and function in the world of molecular biology. Phosphorylation emerges as a critical mechanism in the regulation of DNA packaging, influencing the structure and accessibility of chromatin. This post-translational modification involves the addition of a phosphate group to specific amino acids within histone proteins, effectively altering their interaction with DNA and other nuclear components. By modulating the charge and conformation of histones, phosphorylation can initiate a cascade of structural changes within chromatin. The timing of these modifications plays a pivotal role in cellular processes. Specific kinases are activated during various phases of the cell cycle, facilitating the dynamic rearrangement of chromatin architecture. Such adjustments are essential during DNA replication, where a more relaxed chromatin state permits the replication machinery to access the genetic material efficiently. Conversely, during mitosis, phosphorylation promotes chromatin condensation, ensuring the accurate segregation of genetic material into daughter cells. These modifications are reversible, with phosphatases counteracting the effects of kinases. This creates a finely tuned regulatory system that allows cells to respond to internal and external signals. The balance between phosphorylation and dephosphorylation maintains chromatin in a state conducive to either gene activation or repression. As a result, this regulatory mechanism exemplifies how temporal control of biochemical processes can directly influence gene expression and cellular function, underscoring the complexity of genetic regulation within the nucleus. The cellular environment exerts a profound influence on DNA structure, dictating how genetic material is packaged and accessed. This intricate interplay begins with chromatin, the complex of DNA and proteins that forms the structural basis of chromosomes. In regions where gene expression is active, chromatin adopts a more relaxed conformation, allowing essential transcription factors and RNA polymerases to access the underlying genetic code. This contrast is stark against regions of tightly packed chromatin, where transcription is suppressed. Factors such as histone modifications and the presence of non-coding RNAs contribute to this dynamic restructuring. Chemical signals within the cellular milieu can modify histones, altering their interactions with DNA. These modifications serve as markers that determine whether a gene is accessible or silenced. The influence of neighboring regions also plays a critical role, as enhancers and silencers can communicate over vast distances, affecting the three-dimensional organization of the genome. The mechanical properties of the cellular environment further impact DNA's configuration. For instance, the viscosity of the cytoplasm can affect the movement and interaction of chromatin, influencing its accessibility. This continuous feedback loop highlights how the cellular context shapes the very framework of genetic expression, emphasizing the delicate balance maintained within the nucleus and the broader cellular landscape. The intricate architecture of chromatin plays a pivotal role in the DNA damage response. When DNA strands encounter threats, such as ionizing radiation or chemical agents, this region initiates a cascade of protective mechanisms. Specialized proteins swiftly detect these abnormalities, triggering a series of molecular events that mobilize repair pathways. These pathways are tailored to the type of damage incurred for double-strand breaks. For instance, non-homologous end-joining and homologous recombination serve as primary repair strategies. The former operates rapidly, yet it may induce mutations, while the latter offers greater fidelity and utilizes a homologous template for accurate repair. In this dynamic environment, chromatin remodeling complexes adjust the structure of the chromatin, rendering the damaged sites accessible to necessary repair machinery. This restructuring is crucial, as tightly packed chromatin can hinder the repair process. Additionally, post-translational modifications of histones signify the presence of damage, recruiting repair factors to specific locations on the DNA. Varicula's response to damage is further coordinated by signaling pathways, which ensure that repair processes are activated in a timely manner. Failure in these mechanisms can result in genomic instability, contributing to a spectrum of diseases including cancer. Thus, the DNA damage response within chromatin exemplifies a finely tuned system, safeguarding genetic integrity amidst the myriad of challenges it faces. Telomeres serve as vital guardians of chromatin stability, situated at the ends of linear chromosomes, where they mitigate the loss of essential genetic information during cellular replication. These repetitive nucleotide sequences protect the chromosome from deterioration and fusion with neighboring chromosomes, forming a protective cap. This region is critical, as every replication cycle brings the risk of telomere shortening, a phenomenon that inevitably occurs in somatic cells. As telomeres shorten, they trigger a cellular response that leads to senescence or apoptosis, thus maintaining genomic integrity by preventing the proliferation of potentially damaged cells. The gradual decline in telomere length acts as a biological clock, dictating the lifespan of a cell. In this context, telomeres influence not only individual cellular health, but also the overall longevity of an organism. In some organisms, an enzyme known as telomerase counteracts this shortening, adding repetitive sequences back to the ends of chromosomes. This process is particularly active in stem cells and certain cancer cells, allowing them to bypass the typical limitations imposed by telomere attrition. The interplay between telomeres and chromatin dynamics underscores an intricate balance where the preservation of genetic material is paramount. Understanding this relationship provides insights into aging and the development of age-related diseases, marking a significant frontier in genetic research. Chromosomal territories reveal the intricate organization within the nucleus, where each chromosome occupies a defined spatial region. This spatial arrangement facilitates interactions critical for gene expression and regulation. Within this confined space, specific chromosomes maintain distinct boundaries, minimizing interference from neighboring genetic material. The positioning of these territories is not random, rather, it reflects a complex interplay of molecular signals and structural components. Each territory harbors a unique set of genes and regulatory elements, influencing cellular function. The physical separation of chromosomes aids in preventing gene misregulation, ensuring that cellular machinery can accurately access and utilize the genetic code. Interactions between these territories occur through transient contacts, allowing for coordinated expression of genes located on different chromosomes. This organization is dynamic, responding to cellular needs and environmental changes. During processes such as cell division, chromosomal territories undergo significant reorganization, ensuring accurate distribution of genetic material to daughter cells. Loss of this structural integrity can lead to genomic instability, a hallmark of various diseases, including cancer. Scientific observations indicate that the architecture of the nucleus and its chromosomal territories may also play a role in determining cellular identity. Understanding these relationships enhances knowledge of cellular dynamics, offering potential pathways for therapeutic interventions in age-related and genetic disorders. The cytoskeleton plays a pivotal role in the organization and packaging of DNA within the nucleus. This intricate network of protein filaments provides structural support, maintaining the shape and integrity of the cell while facilitating the precise arrangement of genetic material. Within this region, microtubules, actin filaments, and intermediate filaments interact with chromatin, influencing its compaction and accessibility. Microtubules serve as tracks for motor proteins, which transport chromatin to specific nuclear locations, ensuring that genes are positioned appropriately for transcription and replication. This spatial organization is crucial for gene expression, as the proximity of regulatory elements can significantly impact cellular function. Actin filaments contribute by modifying the physical properties of the nucleus, facilitating transient interactions that enable gene activation. The configuration of this network allows for dynamic responses to cellular signals. When cells divide, the cytoskeleton reorganizes, ensuring that DNA is accurately segregated into daughter cells. Disruptions in cytoskeletal integrity can lead to mispackaging of DNA, resulting in genomic instability and contributing to various diseases. Understanding the cytoskeleton's role in DNA packaging reveals a complex interplay of mechanical and biochemical factors. This knowledge not only enhances insights into cellular function, but also informs approaches to target diseases linked to cytoskeletal abnormalities, opening avenues for innovative therapeutic strategies. Advances in imaging techniques have revolutionized the study of chromatin, revealing intricate details of its architecture and behavior within the nucleus. High-resolution microscopy methods, such as super-resolution imaging, enable scientists to visualize chromatin structures at the nanometer scale, uncovering the dynamic nature of these essential components of cellular machinery. These tools allow for real-time observation of chromatin interactions and modifications, providing insights into the regulatory mechanisms that govern gene expression. The development of fluorescent tagging techniques has further enhanced the ability to track chromatin movements and its spatial organization within the three-dimensional context of the nucleus. This approach sheds light on the role of chromatin in cellular processes such as DNA repair, replication, and transcriptional regulation. By analyzing these behaviors in live cells, researchers can assess the influence of various factors, such as environmental stresses or drug treatments, on chromatin dynamics. In addition to live imaging, advanced techniques like chromatin conformation capture provide a comprehensive view of the spatial relationships between distant genomic regions. This method reveals the folding patterns of chromatin and the formation of loops, which are crucial for understanding how genes are regulated and expressed. Collectively, these imaging advancements contribute significantly to the understanding of chromatin's role in maintaining genomic integrity and its implications in various diseases. This intricate spatial organization of the genome plays a pivotal role in regulating gene expression and cellular function. Three-dimensional arrangements reveal that distant genomic regions can interact, influencing the transcriptional landscape. These interactions are facilitated by the folding of DNA into loops and domains, which create functional neighborhoods within the nucleus. Such organization is not random, it follows specific patterns that are essential for proper cellular operation. Chromatin structural variations contribute to differing gene accessibility. Active genes are often found in open, accessible regions, while inactive genes reside in more compact, repressive zones. This configuration allows cells to efficiently manage the complex demands of gene regulation. Advanced imaging techniques highlight these features, enabling researchers to visualize the dynamic nature of genome architecture in real time. Nuclear compartments emerge as distinct arenas for specific functions. These regions harbor regulatory elements, enhancers, and insulators, fostering intricate networks of gene regulation. The interplay between these compartments can affect cellular responses to external stimuli, offering insights into developmental biology and pathology. Understanding this three-dimensional genome offers profound implications for comprehending diseases such as cancer, where disruptions in spatial organization can lead to aberrant gene expression. The ongoing exploration into this vast and complex landscape continues to illuminate the fundamental principles governing life at the molecular level. Chromatin looping serves as a crucial mechanism by which genomic regions interact, facilitating communication between distant regulatory elements and gene promoters. This phenomenon allows for the spatial organization of DNA within the nucleus, leading to the precise control of gene expression. Within eukaryotic cells, the chromatin structure undergoes dynamic changes, adapting to various environmental cues and developmental signals. The formation of these loops is primarily facilitated by protein complexes that bring together different segments of DNA. These interactions are not random, rather, they are guided by specific sequences and structural features embedded in the genetic material. The resulting architecture creates a three-dimensional framework that influences transcriptional activity, ensuring that genes are expressed at the right time and in the appropriate context. Studies have shown that disruptions in chromatin looping can lead to misregulation of genes, contributing to various diseases, including cancers. The precise positioning of looping structures impacts the accessibility of transcription factors and other regulatory proteins to their target genes, highlighting the importance of these interactions in maintaining cellular homeostasis. Understanding chromatin looping provides insights into the regulatory networks that govern gene expression, revealing the intricate processes underlying developmental biology, cellular responses, and evolutionary adaptations. This area of research continues to expand, offering promising avenues for therapeutic interventions and the treatment of genetic disorders. Transcription factors play a pivotal role in determining DNA accessibility, influencing the intricate processes of gene expression. These proteins bind to specific DNA sequences, facilitating or hindering the transcription of genes. Their presence can modify the chromatin structure, allowing the underlying genetic code to become accessible for transcriptional machinery. This interaction is crucial, as not all regions of DNA are equally accessible. Some remain tightly packed, while others are more relaxed, permitting the necessary access for transcription. The dynamic nature of chromatin remodeling is essential for cellular function and identity. Transcription factors orchestrate this remodeling by recruiting additional proteins that alter the local chromatin environment. These proteins can act as molecular bridges, connecting distant regions of DNA to enhance the transcription process. In this way, transcription factors not only determine which genes are expressed in a given cell type, but also influence the timing and level of expression. The specificity of transcription factors is remarkable, as they can distinguish between similar DNA sequences. This precision ensures that cells can respond appropriately to internal and external signals, maintaining homeostasis and enabling development. Understanding the mechanisms by which transcription factors modulate DNA accessibility reveals insights into the fundamental processes of life and the complexities of genetic regulations. These discoveries may unlock new strategies for addressing genetic disorders, emphasizing the importance of this area of research. Enhancers and promoters serve as pivotal elements in the intricate process of gene activation. These regulatory sequences, found within the vast expanse of the genome, function as critical communication points, orchestrating the expression of genes. Enhancers, often located far from the genes they regulate, possess the remarkable ability to interact with promoters through three-dimensional folding of DNA. This spatial organization enables them to influence transcription factors and initiate the process of gene expression. The language of gene activation involves a complex interplay between these sequences and various proteins. Transcription factors bind to specific regions of enhancers, facilitating the recruitment of the transcriptional machinery to the corresponding promoter. This interaction is finely tuned, allowing cells to respond to a myriad of internal and external signals. The specificity of this system is key. Different cell types utilize distinct enhancers, leading to a unique expression profile. This regulatory precision not only determines cellular identity, but also contributes to developmental processes and responses to environmental changes. Dysregulation of enhancer and promoter activity can result in diseases, including cancer, underscoring their importance in maintaining genomic integrity. Research continues to uncover the nuances of this gene regulation language, illuminating pathways that could lead to innovative therapeutic approaches for various genetic conditions and enhancing our understanding of cellular function. Mediators serve as pivotal components in the intricate network of DNA packaging and gene expression, These proteins facilitate communication between enhancers and promoters, acting as intermediaries that translate external signals into specific genetic responses. Their presence is crucial in the formation of transcription complexes, ensuring that the correct genes are activated or repressed in response to cellular needs. In this context, the spatial organization of DNA within the nucleus becomes significant. Mediators assist in the looping of DNA, bringing distant regulatory elements into close proximity with target genes. This proximity fosters a dynamic interaction, allowing for precise control over gene expression. The influence of mediators extends beyond mere regulation. They also play a role in chromatin remodeling, which alters the structural configuration of DNA. This remodeling can either expose or conceal genetic sequences, effectively determining their accessibility for transcription. The diversity of mediator complexes reflects the complexity of gene expression pathways. Variations in mediator composition can influence cellular responses to environmental changes, contributing to the adaptability of organisms. This adaptability is fundamental, as it not only regulates normal cellular functions, but also has implications in disease states. Understanding the role of mediators in this process continues to unveil the sophisticated nature of genomic regulation and its profound impact on life at the cellular level. The influence of DNA packaging on developmental biology is profound, guiding the intricate processes that dictate cell fate and identity. This organization within the nucleus allows for precise gene expression patterns, ensuring that specific genes are turned on or off in response to developmental cues. Chromatin structure, the dynamic arrangement of DNA and associated proteins, plays a pivotal role in these regulatory mechanisms. In the early stages of embryonic development, the tightly controlled packaging of DNA facilitates the activation of genes essential for differentiation. As cells begin to specialize, the local chromatin landscape transforms, allowing access to necessary genetic information. This process is critical in establishing the diverse range of cell types that form the organisms. Changes in chromatin structure can also lead to variations in gene expression that have lasting impacts on development. Misregulation in these processes may result in developmental disorders or contribute to the progression of diseases. The study of these packaging dynamics provides insights into the fundamental principles of life, revealing how organisms adapt to their environments and evolve over generations.
The interplay between DNA packaging and developmental biology highlights the delicate balance required for normal growth and function. This ongoing research continues to shed light on the complexities of genetic regulation, offering glimpses into the molecular choreography that underpins life itself. Genetic disorders present a fascinating intersection of chromatin structure and function, with deviations from the norm leading to significant health implications. Alterations in chromatin organization can disrupt gene expression, resulting in a range of conditions. These disorders often arise from mutations affecting the proteins responsible for maintaining chromatin integrity. When these proteins fail to function properly, the delicate balance of gene regulation is lost. Certain syndromes illustrate the profound impact of chromatin abnormalities. For instance, a condition known as Rett syndrome is linked to mutations in the MECP2 gene, which plays a critical role in the maintenance of chromatin structure. Disruption of this gene leads to profound neurological symptoms, highlighting the intricate relationship between chromatin and brain function. Another example is the Fragile X syndrome, characterized by an expansion of a repetitive DNA sequence in the FMR1 gene. This alteration affects chromatin configuration, leading to a silencing of the gene and subsequent cognitive impairments. The study of these disorders reveals not only the complexity of genetic regulation, but also the potential for therapeutic interventions that target chromatin dynamics. Research into chromatin structure continues to evolve, with implications for understanding the etiology of various diseases. By elucidating these connections, scientists aim to develop new strategies for diagnosis and treatment, paving the way for advancements in medical genetics. DNA packaging plays a crucial role in cancer biology, influencing not only gene expression, but also the structural integrity of the genome. The compaction of DNA into chromatin allows for the efficient organization of genetic material. But deviations from this tightly regulated system can lead to oncogenesis. In cancerous cells, alterations in the architecture of chromatin often result in the misregulation of genes that control cell growth and division. This misregulation can arise from modifications to histones, offtones, the proteins around which DNA is wound. Changes in these proteins can lead to either excessive gene activation or repression. Uncontrolled activation of oncogenes, combined with the silencing of tumor suppressor genes, creates an environment conducive to tumor formation. In addition, the three-dimensional architecture of the nucleus contributes to the expression of genes relevant to cancer. The spatial organization affects how cells respond to external signals. In malignant cells, this organization can become disrupted, leading to aberrant signaling pathways. Research indicates that understanding the complexity of DNA packaging may unveil novel therapeutic targets. Scientists are investigating ways to restore normal chromatin structure and function, which may inhibit the progression of cancer. Through this exploration, there lies the potential for enhanced treatment methods and improved outcomes for patients battling this multifaceted disease. The evolution of DNA packaging mechanisms reveals intricate processes that optimize genetic material for storage and functionality. Over billions of years, organisms have developed diverse strategies to manage their genetic code, ensuring stability and accessibility. In prokaryotes, such as bacteria, circular DNA molecules are efficiently compacted within the cell through supercoiling, allowing these organisms to thrive in varied environments. Eukaryotic cells, with their complex structures, utilize histones to form nucleosomes. This arrangement creates a higher-order structure known as chromatin. The dynamic nature of chromatin allows for the regulation of gene expression, facilitating responses to environmental changes. These modifications contribute to the organism's adaptability and survival. Within specific regions, chromatin undergoes further compaction, forming tightly packed heterochromatin, which serves to silence gene expression and protect genomic integrity. This multi-layered organization not only safeguards the genetic blueprint, but also orchestrates the delicate balance between accessibility and preservation. In multicellular organisms, the coordination of these packaging mechanisms is essential for processes such as development and cellular differentiation. Epigenetic modifications, which influence how genes are expressed without altering the underlying DNA sequence, avas ans inflias aviati patsmax, play a crucial role in this regulation. Understanding the evolution of these mechanisms provides insights into the fundamental principles governing life and the intricate interplay between genetic structure and function. Comparative genomics reveals that DNA packaging varies significantly across species, reflecting adaptations to diverse environmental pressures and evolutionary histories. In eukaryotes, the presence of chromatin structures is essential for organizing DNA into a compact form that fits within the nucleus. This region of genetic material is not merely a passive holder of genetic information, it is actively involved in regulating gene expression and DNA replication. In simpler organisms, such as bacteria, DNA packaging is entirely different. Their circular DNA sits freely in the cytoplasm, forming a nucleoid. This arrangement allows for quick access to genetic information, facilitating rapid responses to environmental changes. Conversely, Multicellular organisms exhibit complex chromatin configurations where histones and other proteins assist in the winding and unwinding of DNA, thereby controlling access to specific genes. The variations in packaging strategies highlight the evolutionary pressures that shape genetic organization. For instance, in plants, the presence of supercoiled DNA aids in managing the rigidity required for structural integrity, while allowing flexibility for gene expression. In mammals, the dynamic nature of chromatin facilitates the differentiation of cell types, allowing for specialized functions. These differences underscore the adaptability of living organisms, showcasing how DNA packaging is a reflection of both evolutionary pathways and ecological demands. Understanding these variations provides a deeper appreciation for the complexity of life on Earth. Chromatin serves as a fundamental structure within the nucleus, intricately organizing DNA to facilitate essential biological processes. Within these tightly packed regions, it undergoes dynamic modifications, responding to various stimuli, particularly during immune responses. This interaction illustrates a remarkable adaptability, allowing cells to rapidly react to pathogens. Orthogonal and sacral reconfiguration of chromatin grants access to specific genes essential for mounting an immune defense. Enzymes play a critical role in this remodeling, adding or removing chemical tags that regulate gene expression. Such modifications can enhance or suppress the activity of immune-related genes, ensuring a swift and efficient response to threats. This region of genetic material exhibits a keen ability to interpret signals from the cellular environment. When confronted with an invader, it orchestrates a cascade of gene activation, and to the dance of intricatity contrate. The interplay between chromatin structure and immune function underscores the complexity of cellular responses. This relationship not only highlights the importance of chromatin in immediate defense mechanisms, but also illustrates how epigenetic changes can lead to long-lasting alterations in immune memory. Such insights deepen the understanding of health and disease, revealing potential avenues for therapeutic intervention. The influence of aging on DNA packaging presents a complex interplay between cellular integrity and genomic stability. As organisms age, the structural organization of chromatin undergoes significant alterations. This changing architecture can lead to the loosening of tightly wound DNA, resulting in a more open configuration that may increase the accessibility of genetic material to regulatory mechanisms. In this environment, the expression of genes associated with cellular repair and stress responses often becomes dysregulated. The gradual accumulation of epigenetic modifications can impair the precise control of gene activity, contributing to the decline in cellular function. The presence of these modifications may also facilitate the onset of age-related diseases, including various forms of cancer, as the protective barriers that typically maintain genomic integrity weaken. Within this framework, the roles of histone proteins and DNA methylation are critical. These elements, responsible for the chemical tagging of DNA, play vital roles in maintaining the compactness of chromatin and regulating gene expression. Their modification patterns can change dramatically with age, affecting the overall cellular landscape. The consequences of these alterations extend beyond individual cells. They can influence tissue functionality and even organism longevity. This dynamic relationship between aging and DNA packaging underscores the intricate connections that govern the life cycle of cells and their response to environmental factors throughout the aging process. Stem cells exhibit a remarkable capacity for differentiation, which is closely linked to their unique DNA packaging. This specific organization of genetic material plays a pivotal role in determining the fate of these versatile cells. In a pluripotent state, the chromatin is typically in a more relaxed configuration, allowing for the expression of genes essential for maintaining stem cell properties. This open conformation facilitates rapid responses to signaling cues from the surrounding environment. As stem cells begin the process of differentiation, the chromatin undergoes significant structural changes. Regions of the DNA become more tightly packed, effectively silencing genes that promote stem cell identity, while activating those necessary for specific cell types. This transition is not merely a physical reconfiguration, it represents a profound shift in the functional capabilities of the cell. The precise mechanisms governing these alterations are still under investigation, yet they highlight the crucial role of epigenetic modifications in guiding development. In this context, the implications for regenerative medicine are profound. Understanding how DNA packaging influences stem cell behavior could unlock pathways for enhancing tissue repair and developing targeted therapies for degenerative diseases. The relationship between chromatin structure and cell fate may reveal new avenues for manipulating stem cell functions. Environmental stressors play a crucial role in influencing chromatin remodeling, impacting genetic expression in profound ways. Factors such as temperature fluctuations, toxic substances, and nutrient availability can induce structural changes in chromatin, leading to alterations in gene accessibility and transcriptional activity. The dynamic nature of this region allows for rapid responses to external stimuli, facilitating adaptability within cellular environments. Responses to stressors often involve post-translational modifications of histones, which serve to either compact or relax chromatin. Acetylation, methylation, and phosphorylation are among the modifications that can alter the binding affinity of transcription factors, effectively determining whether specific genes are activated or silenced. This adaptability is essential for survival, as it enables cells to quickly adjust their biological functions in response to adversity. The implications extend beyond individual cells, influencing entire biological systems. For instance, chronic exposure to pollutants may lead to lasting changes in chromatin architecture, potentially predisposing future generations to various health conditions. This phenomenon underscores the intricate relationship between the environment and genetic expression. Understanding these processes offers insights into how organisms navigate and survive challenging conditions. By deciphering the mechanisms of chromatin remodeling, researchers can explore potential applications in medicine, agriculture, and biotechnology, harnessing the power of environmental adaptability in innovative ways. Diet plays a crucial role in influencing the epigenetic landscape of organisms, shaping gene expression without altering the underlying genetic code. Specific nutrients and dietary components can modify the chemical tags that regulate chromatin structure, thereby impacting cellular functions. This region of study reveals a complex interplay between metabolism and genetic expression, suggesting that what is consumed has far-reaching effects on health and development. Vitamins and minerals, such as folate and zinc, serve as vital cofactors in the biochemical pathways that donate methyl groups to DNA. Such modifications can silence or activate genes, influencing processes like growth, inflammation, and even susceptibility to diseases. Fatty acids, particularly omega-3 and omega-6, also contribute to the intricate network of epigenetic regulation. Their balance is essential for maintaining optimal cellular functions. Emerging evidence indicates that not only the quantity but also the quality of dietary intake can lead to long-lasting epigenetic changes. For instance, a diet rich in fruits and vegetables may promote beneficial modifications, whereas high-fat or high-sugar diets have been associated with adverse epigenetic outcomes. The implications extend beyond individual health, suggesting that dietary patterns may also influence population-level trends in disease prevalence and resilience. Understanding these connections opens new avenues for preventive health strategies and personalized nutrition approaches. The potential of gene therapy continues to evolve, particularly in the realm of DNA packaging. This intricate process is crucial for the regulation of gene expression and the preservation of genetic integrity. Advances in technology provide insights into how tightly wound structures influence cellular function and the manifestation of diseases. The manipulation of these configurations allows for targeted interventions that can alter gene activity or even correct genetic defect. Innovative methods, such as the use of engineered nanoparticles, are being explored to selectively deliver therapeutic genes to specific cells. These carriers can navigate through biological barriers, ensuring that genetic material reaches its intended destination with precision. The ability to modify the structure of DNA packaging could facilitate safer and more effective treatments for various conditions, including genetic disorders and cancers. Research is ongoing to understand the implications of epigenetic factors that affect DNA packaging. This area holds promise for evolving treatment protocols that adapt to individual genetic profiles, paving the way for tailored therapies. By focusing on the mechanisms of how DNA is organized, scientists aim to unlock new strategies in gene therapy that address not only the symptoms of diseases but their underlying causes as well. The future of medicine may very well depend on our ability to decipher and manipulate the complexities of genetic architecture. CRISPR technology stands at the forefront of genetic manipulation, offering unprecedented precision in altering DNA sequences. This innovative tool has transformed the understanding of chromatin dynamics, allowing scientists to explore how the packaging of genetic material influences cellular behavior. By targeting specific genes, it can modify chromatin structure, leading to changes in gene expression and cellular function. The mechanism involves guiding RNA that directs the Cas9 enzyme to a specific location within the genome. Upon reaching its target, Cas9 induces a double-strand break, prompting cellular repair processes that can be harnessed for gene editing. This interaction not only facilitates gene modification, but also provides insights into how chromatin organization affects accessibility and transcriptional activity. In this context, the ability to reshape chromatin states enhances the understanding of epigenetics, revealing how external factors can influence gene expression without altering the underlying DNA sequence. Modifications to histones and DNA methylation can be studied in real time, showcasing the dynamic interplay between genetic material and its regulatory landscape. To Brasage's progresses, the implications of CRISPR technology extend beyond mere gene editing. It holds promise for unraveling the complexities of inherited diseases and developing targeted therapies. Synthetic biology emerges as a pivotal force in the manipulation of DNA packaging systems. This field integrates principles from biology, engineering, and computational science to create novel biological systems. Researchers harness the power of synthetic biology to design and construct new DNA packaging mechanisms, allowing for more efficient and precise control over genetic material. By employing techniques such as CRISPR-Cas9, scientists can engineer DNA sequences with remarkable specificity. This results in the ability to modify how genetic information is organized and expressed within cells. The reconfiguration of chromatin structures, for instance, can lead to better understanding of gene regulation and expression patterns. Enhanced DNA packaging systems facilitate the study of cellular processes at an unprecedented resolution. These engineered systems also provide insights into the fundamental mechanisms of life. They allow for the exploration of how DNA organization influences cellular behavior, impacting fields ranging from medicine to agriculture. The ability to synthesize DNA with specific structural features opens pathways for creating organisms with tailored traits, potentially leading to innovations in biotechnology. The implications extend beyond mere academic interest. Enhanced DNA packaging systems contribute to advancements in gene therapy, synthetic vaccines, and the development of bio-based materials. This intersection of disciplines fosters a deeper comprehension of life's biological machinery, paving the way for revolutionary applications. The ability to manipulate DNA packaging plays a crucial role in biotechnology applications. This intricacy allows scientists to alter gene expression, enhancing the production of desired proteins. By understanding the mechanisms of chromatin structure, researchers can develop targeted therapies that precisely regulate the genes associated with various diseases. These advancements hold the potential to transform treatments for conditions ranging from cancer to genetic disorders. In synthetic biology, the controlled packaging of DNA serves as a foundation for engineering new organisms with specific functionalities. By redesigning the spatial organization of genetic materials, it becomes feasible to create microorganisms that can produce biofuels or pharmaceuticals more efficiently. These engineered systems reflect a harmonious balance of efficiency and purpose, driven by the underlying principles of DNA organization. This relationship extends to gene therapy as well, where precise packaging can enhance the delivery of therapeutic genes to targeted cells. By utilizing nanoparticles that mimic natural DNA packaging processes, scientists are developing innovative delivery methods that improve the effectiveness and safety of treatments. The intricate balance of accessibility and protection in DNA packaging contributes significantly to the success of these biotechnological endeavors. Understanding these complexities opens new avenues in agricultural biotechnology, enabling the development of crops that thrive under challenging environmental conditions. These advances underscore the pivotal role of DNA packaging in shaping the future of biotechnology. Chromatin presents a compelling target for therapeutic intervention, its intricate structure and dynamic nature offering a wealth of opportunities. The organization of DNA within chromatin influences gene expression, thereby impacting various cellular processes and disease states. Alterations in chromatin modifications can lead to aberrant gene activation or silencing, contributing to the progression of cancers and other diseases. Targeting chromatin can enable the modulation of gene expression through specific compounds that either inhibit or enhance chromatin remodeling processes. Small molecules designed to disrupt the interaction between chromatin and regulatory proteins can restore normal gene function in affected cells. This approach has shown promise, particularly in hematological malignancies, where the reactivation of tumor suppressor genes can lead to significant therapeutic benefits. The development of epigenetic drugs, which modify chromatin structure without altering the underlying DNA sequence, highlights the therapeutic potential within this realm. By understanding the precise mechanisms governing chromatin dynamics, researchers can design more effective treatments tailored to individual genetic profiles. Clinical trials are now investigating various agents that target specific chromatin modifications, aiming to improve treatment outcomes and minimize side effects. Researchers DOM. As knowledge expands about chromatin's role in health and disease, it becomes increasingly apparent that leveraging its complexities could revolutionize therapeutic strategies in medicine. This evolving understanding promises to illuminate pathways toward novel interventions, potentially transforming patient care in the years to come. Advances in computational models of DNA packaging have unveiled intricate details about the organization of genetic material within cells. These models utilize complex algorithms and simulations to predict how DNA strands fold and condense into compact structures, a process essential for cellular function. By mimicking the physical properties of DNA, researchers can gain insights into how these molecules interact with proteins and other cellular components. High-throughput data generation has further enhanced the accuracy of these models. Techniques such as high-resolution imaging and next-generation sequencing provide invaluable information regarding the spatial arrangement of chromatin. This knowledge allows for the fine-tuning of computational approaches, enabling the exploration of how variations in DNA packaging influence gene expression and cellular behavior. Particular emphasis has been placed on the role of epigenetics in shaping these structures. Through the incorporation of epigenetic marks into computational simulations, researchers can better understand how environmental influences affect genetic regulation. The interplay between DNA structure and function is becoming increasingly clear, with implications for development, disease, and evolution. These computational advancements not only deepen our comprehension of fundamental biological processes, but also pave the way for innovative research tools. Such tools could assist in predicting the effects of genetic mutations and in designing targeted therapies, thereby enhancing the precision of medicine. The future of genetic research is poised for profound transformation through these sophisticated computational frameworks. The intricate architecture of DNA relies on a delicate interplay of forces and constraints for efficient packaging within cellular environments. This molecular structure is not merely a linear sequence. It is a highly organized entity that must fit within the confines of microscopic spaces. The double helix arrangement is stabilized by hydrogen bonds and hydrophobic interactions, creating a resilient yet flexible form. These forces play a crucial role in maintaining the integrity of genetic information. Within the nucleus, DNA interacts with proteins, forming complexes known as chromatin. This interaction is essential for regulating accessibility to the genetic code, balancing between tightly packed and loosely organized states. The physical constraints imposed by cellular structures dictate how DNA can be manipulated during replication and transcription. The spatial arrangement of these complexes further influences gene expression, determining which segments of DNA are active or dormant. Mechanical forces also contribute to the dynamic behavior of DNA. Tension and torsion arise during processes such as replication, necessitating molecular machines to relieve stress and maintain structural stability. This tension can influence the folding patterns of the DNA strand, impacting its overall functionality. The balance of these physical properties is crucial, as any disruption may lead to genetic instability and birth. The role of biomolecular condensates in chromatin organization provides insights into the intricate architecture of genetic material. These dense assemblies form through liquid-liquid phase separation, creating transient structures that facilitate the spatial arrangement of chromatin within the nucleus. By concentrating specific proteins and RNA molecules, these condensates contribute to the dynamic regulation of gene expression. Interactions among various components within these regions influence the folding and organization of chromatin. This organization allows for distinct compartments that harbor active or inactive genes, optimizing the accessibility of transcriptional machinery. Enhancer-promoter interactions are enhanced by the presence of these condensates, promoting the formation of transcriptional hubs. The fluid nature of these structures enables rapid reorganization in response to cellular signaling or environmental changes, allowing the genome to adapt efficiently. For instance, during cellular stress, specific condensates may form to sequester and protect essential factors, reducing the burden on the genome and preserving cellular integrity. Research indicates that disruptions in biomolecular condensate dynamics can lead to aberrant gene expression patterns, potentially contributing to various diseases, including cancer. Understanding the mechanisms governing these structures and their interactions with chromatin remains a critical area of investigation, shedding light on the fundamental processes that underpin life itself. Liquid-liquid phase separation plays a pivotal role in the organization of the nucleus. It facilitates the dynamic compartmentalization of various nuclear components, creating distinct environments where biochemical processes can occur with efficiency. These microenvironments are not mere static structures, but rather fluid and responsive, allowing for rapid changes in composition and function based on cellular needs. In this region, specific proteins and RNA molecules undergo phase transitions, leading to the formation of membraneless organelles that serve as hubs for critical activities such as transcription and RNA processing. This phenomenon draws parallels to the behavior of certain liquids, wherein components coalesce under specific conditions, resulting in the demarcation of functional domains. The interplay between these domains is finely tuned by various factors, including concentration, temperature, and the presence of specific binding partners. This intricate regulation highlights the delicate balance that must be maintained within the nucleus to ensure proper cellular function. Observation of these processes reveals insights into how cells adapt to stress, regulate gene expression, and respond to developmental cues. Disruptions in this delicate organization can lead to a variety of diseases, including cancers. Understanding the principles of liquid-liquid phase separation deepens knowledge of nuclear architecture, illuminating the complexities of molecular interactions that drive cellular life. Mechanical forces play a crucial role in the packaging of DNA within the confines of the cell nucleus. These forces arise from various sources, including the cytoskeletal elements, cellular tension, and the inherent properties of the chromatin itself. As the nucleus operates within a dynamic environment, the interplay between these forces and the molecular structure of DNA leads to a sophisticated organization of genetic material. Chromatin, composed of DNA wrapped around histone proteins, exhibits a remarkable ability to respond to mechanical stimuli. Stretching or compressive forces can alter the spatial arrangement of chromatin fibers, affecting accessibility for transcription and replication. This organization is not static. Instead, it is subject to continual remodeling as cellular needs change. The behavior of DNA is influenced by the balance of these mechanical forces, guiding the transition between compacted and relaxed states. This modulation is essential for processes such as gene expression, where the accessibility of certain regions of DNA can dictate whether specific proteins are synthesized. Additionally, insights into the mechanical influences on DNA packaging provide a deeper understanding of how cells maintain integrity under various physical stresses. The ability to adapt to changing environments is pivotal for survival, revealing the intricate relationship between physical forces and genetic regulation within the cellular landscape. This relationship extends to the realm of viruses, where DNA packaging becomes crucial in ensuring successful assembly and replication. The viral genome, often simplified and compacted, must navigate a complex environment to achieve stability while maintaining the necessary information for infection. This precision in packaging underlies the mechanisms that allow viruses to efficiently hijack host cellular machinery. In this context, specific proteins play a vital role, facilitating the encapsulation of genetic material within protein shells. These proteins recognize distinct sequences within the viral DNA, initiating a process that ensures the genome is securely enclosed. This step not only protects the genetic material from degradation, but also prepares it for transmission to new host cells. The dimensions of this assembly process are finely tuned, influenced by the physical properties of the viral genome and the surrounding cellular environment. Any deviation can lead to incomplete or non-infectious viral particles. This necessity for accuracy has led to the evolution of various strategies among different viruses, highlighting the diverse approaches to DNA packaging. As viruses proliferate, they adapt their packaging mechanisms of RNA autodiase, of RNA itabatus. They adapt their packaging mechanisms of RNA autodifrolase, leading to a fascinating interplay between genetic information and the physical constraints of their environment. Understanding these dynamics is essential for developing therapeutic interventions, as it sheds light on the foundational principles that govern viral behavior and host interactions. The interplay between DNA packaging and RNA processing reveals intricate mechanisms that underpin cellular function in eukaryotic cells. The organization of DNA into chromatin directly influences how genes are expressed. This tightly regulated structure determines accessibility, guiding the recruitment of necessary transcription factors and RNA polymerases. When chromatin is in a more relaxed state, genes become more accessible, allowing for efficient transcription into messenger RNA. The process of RNA processing involves several critical steps, including capping, splicing, and polyadenylation. These modifications ensure that the mRNA transcript is mature and ready for translation. The physical arrangement of DNA within the nucleus impacts these RNA processing events. Regions of dense chromatin may hinder the accessibility of transcription machinery, resulting in differential gene expression and impacting overall cellular functionality. Furthermore, the synthesis of non-coding RNAs, such as microRNAs, is also influenced by the structural state of the chromatin. These non-coding molecules play essential roles in regulating gene expression, often acting post-transcriptionally to silence or enhance specific mRNAs. The feedback loop between DNA packaging and RNA processing exemplifies how structural biology intricately intertwines with gene regulation, emphasizing the complexity of cellular life and its adaptive responses to environmental stimuli. Ancestral states of eukaryotic chromatin provide invaluable insights into the evolutionary processes that have shaped complex cellular structures. These early forms of chromatin, characterized by a simpler organization, reveal a time when genetic material was less compact and more accessible. Such configurations facilitated the fundamental mechanisms of gene expression and replication, allowing for rapid responses to environmental changes. In these primordial environments, the interplay between DNA and associated proteins began to evolve, leading to the development of histones and other chromatin-associated factors. The emergence of these proteins marked a significant transition, enabling tighter packaging of genetic material and the formation of nucleosomes. This structural arrangement laid the groundwork for the intricate regulatory networks that characterize modern eukaryotic cells. Through evolutionary pressures, chromatin underwent modifications in both composition and structure. These adaptations allowed for enhanced regulation and specialization of gene expression, which is essential for multicellular organisms. Diverse mechanisms, such as histone modifications and the incorporation of variant histones, contributed to the functional diversity of chromatin. The study of ancestral chromatin states not only illuminates the origins of eukaryotic complexity, but also underscores the versatility of genetic architecture in responding to various evolutionary challenges. Such historical perspectives enrich our understanding of the molecular foundations that govern life as it exists today. Structural biology techniques provide a window into the intricate world of DNA packaging. Advanced imaging methods, such as cryo-electron microscopy, allow researchers to capture high-resolution images of chromatin structures in their native state. This approach reveals the organization of nucleosomes, which serve as the fundamental units of DNA packaging. By utilizing this technique, scientists can observe the arrangement of DNA around histone proteins, shedding light on how genetic material is compacted within the nucleus. X-ray crystallography further enhances this understanding by providing detailed atomic-level insights into the interactions between DNA and associated proteins. This method facilitates the visualization of specific binding sites, illuminating how regulatory elements can influence gene expression through spatial arrangements. Avol-Bastic-Sopatix-Bay enables the study of dynamic processes within these structures. It captures the flexibility and variations of DNA packaging, illustrating how these configurations adapt to different cellular conditions. The integration of these techniques allows for a multifaceted view of DNA packaging, emphasizing the complexity inherent in genetic regulation. Each method contributes unique information, collectively offering a comprehensive understanding of how genetic information is stored, established, and utilized. Investigating these molecular structures continues to reveal the delicate balance between stability and accessibility required for life to thrive. The intricate design of DNA packaging plays a crucial role in gene therapy delivery systems. These systems rely on effective methods to transport genetic material into target cells. The spatial arrangement of packaged DNA determines not only the stability of the genetic material but also its ability to navigate cellular barriers. Various approaches have emerged, utilizing lipid nanoparticles, viral vectors, and polymer-based systems to encapsulate DNA, ensuring optimal delivery. Lipid nanoparticles, for instance, facilitate the transport of mRNA to cells, where it can prompt the production of specific proteins. The self-assembling properties of these nanoparticles enhance their efficiency, allowing for precise targeting. Viral vectors, adapted from naturally occurring viruses, offer another avenue for transporting genetic material. Their inherent ability to penetrate cells makes them valuable tools in gene delivery, although concerns regarding immunogenicity and safety persist. Polymer-based systems present a versatile alternative as they can be engineered to optimize release profiles and cellular uptake. The choice of packaging material is paramount, influencing the release rate of the genetic payload and its subsequent expression. Understanding the mechanistic interactions between these delivery systems and cellular environments paves the way for advancements in gene therapy. Continued exploration of these strategies promises to enhance therapeutic outcomes, potentially transforming the landscape of genetic medicine. The accessibility of chromatin is crucial for the regulation of gene expression and cellular function. This structure, composed of DNA wrapped around histone proteins, dictates how tightly genetic material is packaged within the nucleus. When chromatin is in a more relaxed state, it allows transcription factors and other regulatory proteins to access specific genes, facilitating their expression. Conversely, tightly packed chromatin restricts access, thereby silencing gene expression. Research has revealed that modifications to histones and the DNA itself can influence chromatin structure. These epigenetic changes can be heritable, impacting not only individual cells but entire lineages. Acetylation, methylation, and phosphorylation are among the key modifications that alter the physical properties of chromatin, dictating its accessibility. The interplay between these modifications and the three-dimensional organization of the genome plays a pivotal role in cellular identity and function. In specific cellular contexts, such as during development or in response to environmental stimuli, chromatin remodeling complexes are recruited to facilitate transitions between open and closed states. This dynamic regulation allows for precise control over gene expression patterns. Understanding the mechanisms governing chromatin accessibility opens avenues for innovative therapeutic strategies. By targeting the molecular players involved, it may be possible to modulate gene expression in a controlled manner, potentially addressing various genetic disorders and diseases. This intricate modulation of gene expression hinges on the fundamental importance of DNA packaging. The organization of genetic material within the nucleus is not merely a structural necessity, but a crucial player in evolutionary adaptation. In various organisms, the compactness of DNA can significantly influence cellular function and overall adaptability to environmental changes. Tightly packaged DNA can limit access to specific genes, effectively silencing them under certain conditions, while a more relaxed configuration allows for active transcription in response to external stimuli. This flexibility provides a layer of regulation that has proven vital for survival. Organisms capable of modifying their gene expression patterns in response to stresses often exhibit enhanced resilience. The theme for Ogimpognemue, so unfolds through generations, with advantageous traits being transmitted as a result of these dynamic changes in gene accessibility. Mutations that affect DNA packaging can lead to novel adaptations, allowing species to thrive in diverse environments. This phenomenon is observable across various life forms, from prokaryotes to complex eukaryotes, highlighting the universal significance of how DNA is organized and expressed. The relationship between DNA packaging and evolutionary success serves as a reminder of the delicate balance within biological systems. Understanding these mechanisms can offer insights into the broader implications for biodiversity and the processes that underpin life's complexity on Earth. Chromatin serves as a critical component in the regulation of gene expression, influencing not only development but also behavior. This structure, formed by DNA wrapped around histone proteins, is essential in determining how genes are accessed and utilized. In this context, it plays a pivotal role in behavioral epigenetics, a field that examines how environmental factors can lead to heritable changes in gene expression without altering the underlying DNA sequence. When external stimuli occur, such as stress or changes in nutrition, modifications to chromatin can result. These changes may include the addition or removal of chemical groups, altering the accessibility of specific genes. Such adjustments can lead to variations in behavior across generations, suggesting that experiences can leave a molecular imprint on the genome. In various species, these epigenetic modifications have been linked to adaptive behaviors, enhancing survival in changing environments. Research indicates that offspring can inherit traits shaped by parental experiences, underscoring the dynamic relationship between genetics and environment. This intricate interplay emphasizes that behaviors are not solely determined by genetic blueprints, but are also influenced by the chromatin's responsive nature. Consequently, understanding chromatin's role provides valuable insights into the mechanisms driving behavioral changes, further illuminating the complex web of interactions that define life on Earth. Wonderbothon, integrating systems biology approaches, reveals the multifaceted nature of DNA packaging. This methodology emphasizes the interconnectedness of various biological components. It allows researchers to explore how chromatin structure influences gene expression, cellular function, and data from high-throughput sequencing technologies further enrich this understanding. By mapping DNA-protein interactions and identifying regulatory elements, researchers uncover patterns that govern genetic information's accessibility. Each layer of complexity, from epigenetic marks to three-dimensional genome architecture, reflects the finely tuned mechanisms that sustain life. Disciplinary collaboration enhances this exploration. Biologists, physicists, and computer scientists pool their expertise to analyze vast datasets, leading to new hypotheses about genetic regulation. In this context, the study of DNA packaging emerges as a crucial piece of a larger puzzle, revealing not only how genes are organized but also how they interact with environmental cues and internal cellular pathways. The implications extend beyond basic biology. Insights gained from these systems approaches inform medical research, potentially guiding targeted therapies for genetic disorders and cancer. Thus, the integration of various scientific disciplines fosters a deeper comprehension of life's fundamental principles and challenges the boundaries of current knowledge. Genetic research continues to evolve, revealing complexities in DNA packaging that reshape our understanding of genetics. This intricate system governs gene expression, influencing how cells interpret genetic information. With advancements in technology, researchers can now visualize these processes at unprecedented resolutions, allowing for a detailed exploration of chromatin structure and its dynamic nature. The role of epigenetics extends beyond mere genetic regulation. Environmental factors, lifestyle choices, and cellular conditions can induce modifications that affect gene activity. These alterations may not only impact the individual, but can also be passed down through generations, suggesting a profound connection between ancestry and contemporary biological functions. In this context, the study of DNA packaging is critical. Histones and other proteins play pivotal roles in organizing DNA into compact structures, thereby regulating accessibility to genetic material. The unraveling of these mechanisms has significant implications for understanding diseases, particularly those with genetic underpinnings, such as cancers and hereditary disorders. Future research aims to decipher the epigenetic code, which holds the potential to unlock new therapeutic avenues. By targeting specific pathways, scientists may develop interventions that reverse detrimental epigenetic modifications. This trajectory promises to not only enhance our understanding of genetic regulation, but also to address health challenges that arise from complex genetic interactions. The role of DNA packaging in plant biology reveals intricate mechanisms essential for growth and adaptation. Within these organisms, DNA is meticulously organized into structures known as chromatin. This regulation allows for the precise control of gene expression, which is critical in response to environmental stimuli. Certain regions of chromatin are tightly packed, limiting access to the underlying genetic material. This compaction serves to silence genes, while more loosely arranged segments foster active transcription. Different plant species exhibit variations in their chromatin architectures, adapting their genetic responses to diverse climates and ecological niches. For instance, in arid environments, specific genes may be upregulated to enhance drought resistance. The chromatin landscape shifts dynamically as plants encounter varying conditions, reflecting their remarkable resilience. Epigenetic modifications further influence the packaging of DNA, allowing plants to remember past environmental stresses and adjust their growth patterns accordingly. Such mechanisms involve the addition of chemical groups to histones, altering chromatin structure without changing the underlying DNA sequence. This adaptability underlies the evolutionary success of many plant species across the globe. Understanding these processes opens avenues for biotechnological applications, including crop improvement and sustainable agriculture. By manipulating DNA packaging, researchers can enhance traits like yield and stress tolerance. Theoretical models of DNA packaging introduce a fascinating intersection of biology and mathematics. These models seek to decode the complex arrangements that enable efficient genetic material storage within the confined spaces of a cell nucleus. Utilizing geometric principles, researchers explore how long strands of DNA can be compacted into structures that occupy minimal volume while maintaining accessibility for transcription and replication. Mathematical frameworks often employ concepts from topology and combinatorial optimization. By simulating various configurations, scientists can predict the most stable forms that DNA might adopt under different cellular conditions. This includes the exploration of nucleosome positioning and the three-dimensional folding patterns that influence gene expression. These geometric arrangements are not merely abstract. They have profound implications for cellular function and development. The structure of DNA can affect how genes are turned on or off, influencing everything from cellular morphology to an organism's response to environmental stimuli. The intricate nature of this packaging is governed by both physical forces and biochemical interactions, resulting in a dynamic system that adapts over time. This region of study continues to evolve, revealing layers of complexity that challenge existing paradigms and offer new insights into the molecular mechanisms underlying life itself. Each discovery contributes to a broader understanding of genetic regulation, bridging gaps between mathematical theory and biological reality. The intricate arrangement of chromatin plays a vital role in maintaining genome stability. This region, characterized by its complex folding and organization, serves as more than a mere structural framework. It influences how genetic material is accessed and utilized. Variations in chromatin structure can lead to the exposure of sensitive sequences, heightening the risk of mutations or genomic instability. Histones, the proteins around which DNA wraps, contribute significantly to this architecture. Their modification and interaction determine the accessibility of certain genes, creating a dynamic environment where expression and repression are finely tuned. Changes in histone modifications often correlate with shifts in chromatin states, affecting the overall stability of the genome. When chromatin becomes disorganized, the repercussions can be severe. Misfolding or aberrations in this structure may result in double-strand breaks, which pose a threat to cellular integrity. Repair mechanisms, while robust, can be overwhelmed when faced with extensive damage. The interplay between chromatin and DNA repair pathways illustrates a finely balanced relationship. Additionally, regions of repetitive sequences, often found within heterochromatin, present unique challenges. Their propensity to form secondary structures can complicate replication, further testing the resilience of the genome. Understanding these complexities remains crucial to unraveling the profound implications of chromatin structure on cellular health and stability. The influence of chromatin accessibility on disease susceptibility extends the conversation into the realm of genetic expression and regulation. This region, where the tightly packed DNA resides, plays a pivotal role in determining how genes are accessed and activated. When chromatin is densely packed, it restricts the ability of transcription factors to reach their target sequences, limiting gene expression. In contrast, regions of open chromatin allow for greater accessibility, facilitating the transcription of genes that can impact cellular function and overall health. Variations in chromatin structure can contribute to an individual's vulnerability to diseases. Certain conditions, such as cancer, arise when regulatory mechanisms fail, leading to aberrant gene expression. This misregulation often stems from altered chromatin states, which can be induced by environmental factors, lifestyle choices, and intrinsic genetic mutations. The relationship between chromatin accessibility and disease is increasingly evident in autoimmune disorders and neurological conditions. The dynamic nature of chromatin allows it to respond to external stimuli, but this flexibility also means that it can become dysregulated, triggering pathological states. Researchers continue to explore how the manipulation of chromatin structure could offer avenues for therapeutic intervention, potentially restoring normal gene expression patterns and enhancing cellular health. Each discovery further illuminates the intricate interplay between genetic architecture and disease. The role of DNA packaging in regenerative medicine is pivotal in understanding cellular behavior and therapeutic advancements. This region of research investigates how the folding and compaction of genetic material influence cellular identity and function. The organization of DNA within the nucleus dictates accessibility to genetic information, which, in regenerative medicine, manipulating DNA packaging can enhance the efficiency of stem cell therapies. By modifying the chromatin structure, researchers aim to create environments that favor the expression of specific genes essential for tissue repair. This approach involves employing small molecules or proteins that can alter the state of chromatin, promoting a more favorable configuration for gene activation. The implications of these findings extend to the treatment of degenerative diseases, where restoring cellular function is paramount. Innovations in gene editing technologies, such as CRISPR, are being optimized to work alongside these packaging strategies, ensuring precise modifications that support regeneration. The targeted delivery of therapeutics relies heavily on understanding the spatial organization of DNA, allowing for a more effective approach to healing damaged tissues. As regenerative medicine continues to evolve, the interplay between DNA packaging and cellular mechanisms promises to unlock new pathways for treating previously incurable conditions. This ongoing research holds the potential to redefine the boundaries of medical science, offering hope for future therapies. Emerging technologies are reshaping the understanding of chromatin dynamics, enabling scientists to observe the intricate packaging of DNA in unprecedented detail. Techniques such as single-molecule imaging and high-throughput sequencing facilitate real-time monitoring of chromatin structures as they respond to various stimuli. This new capability allows for the examination of how genetic material is organized within the nucleus, revealing the fluid nature of these arrangements. Advanced microscopy techniques, including super-resolution microscopy, provide insights into the spatial organization of chromatin at a nanometer scale. These methods uncover the complex interactions between DNA, histones, and regulatory proteins, highlighting the importance of epigenetic modifications in gene expression. The use of CRISPR technology enables targeted editing of chromatin structures, allowing researchers to manipulate DNA packaging and observe resultant changes in cellular behavior. Integration of artificial intelligence in this field further enhances data analysis, offering algorithms that can predict chromatin behavior based on large datasets. These innovations pave the way for identifying potential therapeutic targets in various diseases, including cancer, where chromatin alterations play a significant role. The continuous evolution of these technologies promises to deepen the understanding of genetic regulation and its implications. The significance of DNA packaging extends into the realm of forensic science, where the organization of genetic material plays a crucial role in the analysis of biological evidence. In this context, the compact structure of DNA, facilitated by histones and chromatin, enhances the efficiency of extraction and amplification processes. This organization allows for the preservation of genetic integrity, even in degraded samples commonly encountered at crime scenes. Genetic packaging mechanisms enable forensic scientists to distinguish between different genetic profiles with remarkable accuracy. This precision is essential when analyzing trace evidence, such as hair follicles or skin cells, which can contain only minute quantities of DNA. The spatial arrangement of nucleosomes and the accessibility of specific genes contribute to the reliability of polymerase chain reaction techniques, which amplify targeted segments for further examination. The ability to interpret packaged DNA not only aids in identifying individuals, but also provides insights into familial connections and patterns of inheritance. These insights can illuminate relationships between suspects and victims, expanding the scope of investigations. Advanced methods of DNA packaging analysis also enhance the understanding of epigenetic modifications, which can influence gene expression and thus offer additional layers of information regarding an individual's background. Such comprehensive approaches underscore the pivotal role of DNA packaging in forensic science, revealing its far-reaching implications in the pursuit of justice. The societal implications extend beyond the realm of forensic science, shaping perspectives on identity, ancestry, and the intricate web of human relationships. Knowledge of DNA packaging informs the understanding of genetic predispositions, an insight that could guide medical interventions and public health strategies. Such understanding has the potential to alter individual behaviors, encouraging proactive health measures and personalized treatments based on genetic information. This region of genetic inquiry also raises ethical questions. The distinction between genetic determinism and free will becomes increasingly blurred as individuals grapple with the knowledge of their hereditary traits. Families may face complex decisions regarding testing and the sharing of genetic information, impacting dynamics and social structures. Concerns over privacy and the potential for discrimination loom large, as access to genetic data could lead to biases in employment, insurance, and other societal sectors. Balancing the benefits of DNA understanding with the ethical dilemmas it presents is crucial. Educational initiatives can foster informed discussions, equipping communities with the knowledge needed to navigate these challenges. Within this evolving landscape, the implications of DNA packaging resonate deeply, touching on cultural narratives and the human experience itself. The ongoing exploration of genetic architecture continues to inspire dialogue, urging society to consider the profound intersections between science, ethics, and humanity. Questions regarding ethical considerations in genetic manipulation arise as advancements in DNA packaging techniques evolve. This region of study delves into the moral implications of altering the genetic code, highlighting the potential for both remarkable benefits and unforeseen consequences. The capability to manipulate genes may lead to breakthroughs in medicine, agriculture, and biotechnology, yet it simultaneously invites scrutiny over the inherent risks. Concerns emerge surrounding the unintended impacts on ecosystems and biodiversity. Manipulating genetic structures could create imbalances, affecting not only targeted species but also broader ecological networks. The delicate interplay between organisms necessitates a careful examination of the long-term effects of such modifications. Ethical frameworks must address the consequences of these actions, especially when considering the potential for engineered organisms to outcompete or disrupt natural populations. Furthermore, the societal ramifications of genetic manipulation prompt important discussions. Issues of accessibility and inequality may arise as advanced treatments become available only to certain segments of the population. The potential for genetic enhancements raises questions about the definition of normalcy and the societal pressures that may accompany these innovations. As the field progresses, balancing scientific advancement with ethical responsibility remains crucial, guiding researchers and policymakers in navigating the complex landscape of genetic manipulation and its implications for future generations. The trajectory of DNA packaging research holds immense potential for various scientific fields. Innovations in understanding how genetic material is organized within the nucleus, could lead to groundbreaking discoveries in biotechnology and medicine. Insights gained from exploring chromatin structure and histone modifications may transform gene therapy approaches, providing precise methods to target genetic disorders at their source. In this evolving landscape, the implications extend beyond immediate scientific applications. The refinement of genetic editing technologies, such as CRISPR, hinges on a comprehensive grasp of DNA architecture. This knowledge not only informs effective editing strategies, but also enhances the accuracy and safety of such interventions. Collaboration among biologists, chemists, and computational scientists is vital in pushing the boundaries of this research. Interdisciplinary efforts could yield sophisticated models that simulate DNA dynamics, enabling predictions about cellular behavior under various conditions. Public engagement and education will play significant roles in shaping perceptions of genetic research. Fostering an informed dialogue between scientists and society will help navigate the ethical considerations surrounding these advancements. As the understanding of DNA packaging deepens, the capacity to manipulate biological systems responsibly will likely expand. The future of this research promises a nuanced interplay between innovation and ethical stewardship, steering the course of scientific discovery into uncharted territories.